Hello and welcome to our second genetics lecture. Today we'll be talking about more complex genetics, situations where the genetics of traits is more complex than what we talked about previously. Genetics can get extremely complex, and there's upper division classes that are only on genetics. So even though we're talking about stuff that's a little more complex, we're still really only looking at the tip of the iceberg. Alright, so we are going to go through 4 examples where we're going to build on what we learned from the last genetics lecture and look at these cases that are just a little more complex. So first we'll look at sickle cell disease, and then we'll look at blood types, eye color, and then cat fur color. Alright, so the first is sickle cell disease. Hopefully you remember that sickle cell disease, or SCD, is caused by a mutation in the hemoglobin gene, and it causes red blood cells to become sickle-shaped, which then gives all sorts of complications to the person who has that. So we've looked at the mutation in the DNA and how it changes the primary structure of the protein, which changes the overall shape of the protein, which then changes its function. Okay. If you don't recall that stuff from previous lectures, I suggest you go back and look at those again. So now we're going to talk about the genetics of sickle cell disease. So how do you get sickle cell disease? Well, first of all, it is an inherited autosomal trait. Okay, so inherited means that you get it from one of your parents who inherit it, so it has a genetic cause. Autosomal is a term that means that it is a gene that is not on a sex chromosome. It is not on the X chromosome. It is on one of the 22 autosomes. Okay. So there is one gene for the beta globin subunit of hemoglobin, and there are 2 alleles for this gene. Okay. There's the normal hemoglobin allele, and we call this hemoglobin A, and the notation we use is H superscript A. Okay, so that's for the normal non-mutated version of the hemoglobin, and it's the beta globin subunit. Okay. And then there's the sickled hemoglobin, which we also call hemoglobin S, and the notation for that is H superscript S. Okay, that's the mutated version of the hemoglobin that causes, that causes this situation. Okay, this is interesting because it is an example of codominance that's observable only at the molecular and cellular level. So you can't see it when you look at somebody. You have to be able to look at their red blood cells or genotype them. All right, so here's a chart that has all sorts of information. So in this column lists the genotype. Okay, remember each person is going to have 2 copies of the gene. Okay, and they could have 2 copies of the same allele, or they could have different alleles on their 2 copies. And then we're going to look at the types of hemoglobin that are made by those people with each genotype, what their red blood cells look like, and talk about whether or not they have sickle cell disease. Okay, so for starters, let's look at someone who is HA. This means they have only normal copies of the allele. They don't have a mutated version of that allele, so they will only make hemoglobin A. Okay, all of their red blood cells will be nice and round and normal, and of course this person would not have sickle cell disease. Okay, let's skip that for a moment and look at someone who would be homozygous for the sickle cell, so for the sickled hemoglobin, excuse me. So 2 copies of the sickled hemoglobin, and that person would only make hemoglobin S. Okay, and all of their red blood cells would be sickle shaped, and this person would have sickle cell disease. Okay, those make sense, right? Okay, now let's look at this one, the people who are heterozygotes. So they have 1 copy of the normal allele and 1 of the sickled hemoglobin allele. Okay, they make both hemoglobin A and hemoglobin S. Okay, and that means that some of their red blood cells will be nice and round and some will be sickled. Okay, these people don't have the disease, they don't show symptoms, or if they do, they're very, very mild symptoms, or only triggered by low oxygen levels, like if they go really high up in the mountains. Okay, so they don't have the disease because they have plenty of these round red blood cells with hemoglobin A, and they're functioning just fine, and it's only under certain circumstances. Okay, so here we see that the two alleles, the H superscript A and H superscript S, are codominant, right? And we see that because both types of hemoglobin are made, right? And then we get both types of red blood cells. If A was dominant over S, if HA was dominant over HS, then we would see that these heterozygotes would just be normal, right? Or if it was incomplete dominance, then you would have some sort of mixing or part of normal and part sickle cell. But it's not, it's codominant, okay? So remember that the heterozygotes have phenotypes of both alleles, so we see that here. And we call these carriers, okay? So people who have one of the disease alleles and one of the normal alleles are carriers of this disease. They don't have it themselves, but they can give it to an offspring, okay? And they have a 50% chance of passing that diseased allele onto offspring. Okay? We also talked about carriers when we talked about sex-linked genes or sex-linked diseases, and that term also applies to women who have one copy of a disease allele and one copy of a non-disease allele on their X chromosome. Okay, so the whole idea with a carrier is you don't actually exhibit that disease or that phenotype, but you carry the gene or carry the disease allele. All right, so that takes care of it for sickle cell disease. Okay. Next are ABO blood types. Okay, this also is an inherited autosomal trait. Okay, and blood type is controlled by one gene. Okay, everybody has a blood type. So everybody has this gene, and we have two copies of that gene, right, because we're diploid. The gene that controls what blood type you have actually controls for an enzyme that attaches carbohydrates to membrane proteins on the surface of red blood cells. And we call these proteins glycoproteins because they have part sugar and part proteins. Okay? And their whole purpose is self-identification. So here's the diagram of the cell membrane that we had in our cell membrane lecture way back. And this here is a glycoprotein, and it has both the protein part and the carbohydrate part, and so that's what we're talking about. So there's on the red blood cell surface, there's these proteins sticking out with different carbohydrates. Your immune system can recognize what version you have, and they see that, and your immune system recognizes that as your own blood, right? It's really important because you don't want your immune system to attack your own blood. That would cause death. All right, so for this one gene that makes this makes the enzyme that makes the glycoprotein, there are three alleles. Okay, there's the A allele that makes glycoprotein A, so version A. Okay, it's the protein with a particular carbohydrate attached to it. Okay, there's also the B allele, which codes for an enzyme that makes a slightly different version of the carbohydrate, so it makes a different version of the glycoprotein, called glycoprotein B. And then there's the O allele. The O allele is a mutated version of that enzyme, and it's a non-functional, and so this codes for no glycoprotein. Okay. So this is interesting because it's an example of multiple alleles. Okay, so we're dealing with three alleles here. Everything else we have talked about before, there's only two alleles. Okay? And this is also an example of codominance that is observed only at the molecular level. All right, so these three alleles give rise to four different blood types, and the blood type here is the phenotype. Okay, so the phenotype, of course, is what you exhibit, right? So each one of us has a blood type, and everyone has to be one of these blood types: type A, type B, type AB, or type O. Okay. So how do these three alleles give rise to the four blood types? Okay, so it all has to do with how the alleles interact with each other. Okay, so the first thing you need to know is that the O allele is recessive to both the A and the B alleles. Okay, this makes sense because remember the O allele makes a non-functional enzyme. Okay, so if you have the O allele, it's saying don't make any enzyme. Okay, and if you don't make any enzyme, you're not going to have any carbohydrate being made. Okay, but the A allele is saying make the A carbohydrate, and the B allele is saying make the B carbohydrate. Okay, so not making any carbohydrate is recessive to both of those. And then A and B are codominant to each other. Okay, so here's another chart. This is listing the phenotype, which in this case is the blood type. And then we're going to think about what glycoprotein is made, what the red blood cells look like, and what genotype or genotypes go along with these phenotypes. Okay, so first of all, for type A blood, the glycoprotein that will be produced is glycoprotein A, right? And the red blood cells will look like this. Okay, these triangles are supposed to represent glycoprotein A. Okay, so the red blood cells will have a bunch of these glycoproteins sticking out on the surface. Okay, and that person's immune system would recognize that as, hey, those are my blood cells. I shouldn't destroy them. Okay. The genotypes that go with this, there are actually two. Okay, so remember you have the A allele, the B allele, and the O allele, and each person is going to have two of those. Okay, so definitely somebody who makes glycoprotein A, they must have one copy of the big A allele, right? So they could be homozygous for the big A allele, and they would have blood type A. But there's another possible genotype, and that's AO. Okay, because remember A is dominant over O. Okay, so someone who is AO will still make glycoprotein A because one of their alleles will say to make that. And then the O allele just says don't make anything. So, it's like the null. Okay. Okay, the next is blood type B. Okay, and this is going to I'm going to abbreviate glycoprotein GPB. Okay, so a person with blood type B makes only glycoprotein B. And the red blood cell could be represented like this, with these little balls representing glycoprotein B on the surface of the red blood cell. Okay. Notice they don't make A, just like this one doesn't make B. Okay, so someone who's only making glycoprotein B must have one copy of the B allele. Okay, and just like with the A, type A blood, they could be homozygous for that allele, or they could be heterozygous with the recessive allele. Okay, for the same reason. Someone who has just one copy of the big B allele will still have the glycoprotein B, regardless of whether or not they have a B or an O on their other allele. All right, so those ones are pretty straightforward. Now let's think about this one. So someone who is type AB, that means that they are making both glycoprotein A and glycoprotein B. Okay? And their red blood cells would show a combination of those, right? So there's glycoprotein A and there is glycoprotein B. So the red blood cells would have both versions on them. And there's only one genotype that's possible to go along with type AB blood, and that is the genotype of AB. Okay, so one of their alleles has to be saying make glycoprotein A, and the other allele must be saying make glycoprotein B. Okay, remember, you just have two alleles, so you can't have the O in there at all. Okay, these, of course, are the heterozygotes. Okay, and they have one, different alleles, and that shows that these are codominant. Why are they codominant? Well, because when you look at the red blood cell, you see both of them. You see the A glycoprotein and the B glycoprotein. Okay, and then finally there's the type O blood. So someone who has type O blood doesn't make any glycoprotein. So not glycoprotein A or glycoprotein B. Okay, so in other words, they make no glycoprotein. And so their red blood cells, as far as this glycoprotein is concerned, are naked. They don't have any self-identification factors on them. Okay. This is the case. This is the recessive allele. Remember, O is recessive. The O allele is recessive to both the A and the B alleles. So someone who has type O blood must be little o, or must be OO, must be homozygous recessive. Okay. So usually when I'm teaching this in class, people ask me a couple things. So first of all, they ask about, oh, I should have just put in an extra piece of thing, but I didn't. Okay. So they ask about the negative or the positive thing, right? So when you get your blood typed, you'll be told that either you have, something like type A negative blood or you're type O positive. So they're actually telling you two things. They're telling you what your blood type is for the ABO blood type, and then they're also telling you something about a different gene, which is the rhesus factor gene. I'm not going to talk much about that, but I just want to tell you that it's a separate gene on top of that, and it either you have a version that says make this other factor or protein that goes on your red blood cells, or don't. Okay. The other thing people are usually curious about is about blood donations. Okay, so when you receive blood from somebody else, you have to make sure that the blood matches your blood so that you don't launch an immune response against the donated blood. Okay, so for someone who has type A blood, they can, I'm going to write the donations, that's an ugly line. I'm going to try to make a nice straight line down here. Okay, so I'll write the donations who can donate to this person over here. So someone with type A blood, they could be AA or AO, but they need to have blood that either doesn't have anything on it, right, because that won't trigger an immune response. Remember, this person recognizes the A glycoprotein. It would, as self. It would recognize the B glycoprotein as an enemy and launch an immune response. Okay, so someone who has type A blood can get blood from someone who is AA or AO or OO, right? So these both have the same type A, right? That's type A blood and that's type O blood. Okay, similarly, someone with type B blood could get a donation from someone that has type B blood as well, and that would be BB or BO person, or also someone who is OO. Okay. Someone who has type AB blood, now they're not going to recognize either the B allele or the A allele as an enemy. So that means they can actually get blood from anybody. They can get blood from someone with type B blood, who is BB or BO, someone with type A blood, who is AA or AO, someone also with AB blood, and then also someone with type O blood. And then what about this person who's type O? Type O blood is going to not recognize any glycoprotein as self, and so if it gets any, the person's body would launch an immune response, and that means the only person, or the only blood type that can donate to a person with type O blood is also a type O. They'd have to be OO. Okay? So what you notice here, hopefully, is that we keep seeing this genotype, OO, which goes with the phenotype type O, over and over again, right? So someone with type O blood is a universal donor. That means they can give blood to anybody else, okay? And it's because they have these red blood cells that don't have any glycoprotein on them. Okay? So we call that the universal donor. The other thing that you might notice is that this person who has AB blood can get blood from anybody else, okay? And so we call that the universal acceptor. So someone who has type B blood, is got it made because they can get blood from anybody. Okay? So luckily for these type O people that can only get blood from other type O people, type O blood is the most common. Okay, so there's usually enough people around with type O blood. Alrighty, let's change gears and talk about human eye color. Okay, so there are 3 basic eye colors, and the eye color is the phenotype. Okay, there are brown eyes, green eyes, and blue eyes. Okay, so obviously this is an oversimplification of eye color, right? So we talked about brown eyes, talked about green eyes and blue eyes, but what about these other ones that are in between, like light brown or grayish or hazel, or ones that have flecks of different colors in it? So how eye color is determined is still being worked out by geneticists, and we don't know everything about it. So we do understand brown eyes, green eyes, and blue eyes, so we're going to talk about that. Okay, so first of all, eye color is the result of the amount of melanin that's produced in the iris of the eye, right? So that's this colored part out here. That was a not very good circle, but this colored part out here, that's the color of your eye, okay? So how much melanin is produced in those cells of the eye, of the iris, determines what color your eyes are. Okay, so let's start down here with brown eyes. So this is supposed to be showing a brown eye, and then this is supposed to be a cell that's part of the iris, and it's called a melanocyte. Okay. A melanocyte is a cell that produces melanin, which is a pigment, which is a brown pigment. Okay. So someone with brown eyes has many melanocytes producing lots of melanin in their iris. Okay. Someone with green eyes has fewer melanocytes producing fewer melanin, and then someone with blue eyes has either no melanocytes or very few melanocytes, not producing very much melanin at all. Okay. So there are 2 things, right? There's the number of melanocytes, that a person can have in the iris of their eye, and then in those melanocytes, how much melanin is produced can vary. All right, so like I said, we don't know everything about how eye color comes about, but we do know that the amount of melanin produced in the iris is determined by at least 2 genes. Okay, probably more, but people have worked out these 2. Okay, and these 2 genes are called, one, the OCA2 gene. Okay, so that's one gene that controls the amount of melanin, and the other one is called the GAY gene, G-E-Y gene. Okay, and that one also controls the amount of melanin. Okay. This is interesting because it's an example of what's called polygenic inheritance, and this is when one trait is influenced by multiple genes. Okay, so the trait we're looking at here is eye color. Okay, and it's controlled by at least 2 genes, the OCA2 gene and the GAY gene. Okay, possibly, probably more as well. No. I gotta keep doing this to myself, writing over areas that Okay. So the OCA2 gene and the GAY gene. Okay, each one of these has 2 alleles. Okay, so we're talking about 2 separate genes here. Everything else that we have talked about up until now, we've only been talking about 1 gene. Okay, we've talked about 1 gene that has 2 alleles. We've talked about 1 gene that has 3 alleles, but we haven't ever talked about how 1 trait can be influenced by 2 separate genes. Okay, so that means that these can be on separate chromosomes, and that they're completely different genes with their own, sequence of bases and their own protein product, right? Okay, but each have 2 alleles. So for the OCA2 gene, there's a brown allele, that's the big B, and also a blue allele, the little b, represented by the little b. Okay? And with the GAY gene, there are 2 alleles. One is the green allele, represented by a big G, and then also a blue allele, represented by a little g. Okay? Even though these have the same name, they have different letters, because they're actually different genes. Okay, and so it's the interaction between these 2 genes and which alleles are present that result in these 3 basic eye colors. And the ticket to understand that, this whole thing, how these interact, is that one, the big B brown is dominant over everybody else. So dominant over little b, dominant over big G, and dominant over little g. Okay, so that means in order to have, if you have this big B allele, you just need one copy of it and you're going to have brown eyes. Okay? And then in addition to that, the G allele, big G allele, is dominant over the little b and the little g. Okay, so big G is dominant over little b and little g. Okay, so like in the hierarchy, it goes big B, then big G, then little b and little g together. Okay, so we're going to look at how these two genes interact with the alleles that are present to form the three different basic eye colors. Okay, so for the brown eyes, okay, so if you have the brown eye phenotype, what does the brown eye genotype have to be? Okay, so we have to consider both the OCA2 gene and the gay gene. Okay, for the OCA2 gene, it has to have at least one copy of the big B allele. Okay, and then the second allele could be a big B or a little b, right? So that means because big B is dominant over the little b, big B, and big B, little b are both going to exhibit brown. Okay, but then you also have to consider the gay gene. But actually, for brown eyes, this is completely irrelevant because remember, the big B is dominant over everybody else. So for the gay gene, you could have big G, or you can have big G, or you could even have little g, and it doesn't matter as long as you have one big copy of the big B allele. And that's because big B is dominant over everybody else. Okay? So here are the possible genotypes for someone with brown eyes, right? Notice what these have in common is that each one of them has at least one big B. Okay? And this just lists every other combination of those other three alleles, right? So we have big B, big G, big B, little b, big G, and so on. Okay? If this isn't making sense to you, so I've combined these two with each one of these, and then these two with each one of these to get those 6 kinds. Okay? If that's not quite making sense to you, you might want to sit down and work it out for yourself. Okay? So this genotype is considering both of those genes, right? And for both of those genes, you have 2 copies of that gene, so you have to list it. You have to list the alleles for both of them. Okay, so this is pretty interesting. We have 6 different genotypes, but they all result in one phenotype. All right, let's look at the same thing for the green eye phenotype and genotype. Okay, for the OCA2 gene, in order to have green eyes, you must be homozygous recessive for the OCA2 gene. So you must be little b. And that's because the presence of a big B would mean brown eyes, right? So you have to have the little b, you have to have 2 copies of the little b. Okay, and then for the gay gene, you have to have at least 1 copy of the big G allele. Okay, and the other allele could be big G or little g, right? And that's for the same reason as when we were talking about the brown eyes. So both big G and big G, little g are both going to express green eyes because big G is dominant over little g and dominant over little b. Okay, so possible genotypes for a green-eyed person are little b, big G, or little b, big G, little g. Okay, notice that these both have a big little b, and then both have at least 1 copy of the big G allele. Okay. So here we have 2 genotypes and 1 phenotype. Okay, and then finally let's look at the blue eye genotype. Okay, so for the OCA2 gene, a person who has blue eyes must be homozygous recessive, right? And this for the same reason as with the green eyes. If there was a big B present, that person would not have blue eyes, they would have brown eyes. Okay? And then same thing for the gay gene. The person must be homozygous recessive For the gay gene also, there must be little g, and that's because if they had a big G, they'd have green eyes. Okay? So there's only one possible genotype for a person with blue eyes, and that's that they must be little b, little g, or homozygous recessive for both genes. All right, now we're going to go on and talk about cat fur color. This is our last example. Okay, so cat fur color is controlled by multiple genes. When you look across the board at different cat colors, cats come in all sorts of colors and patterns. The genetics of how the fur looks is actually really complex. So there's a gene that controls whether or not the cat is tabby or not, so that's showing a tabby cat. There's a gene that codes for color, whether it's black or orange. There's a gene that codes for distribution of color, so whether or not the cat has white patches or is solid. Right? And then there's a situation where we have calico cats. Calico cats have 3 different colors, and they may or may not have patches of white. Usually when they're like this, well, like the whole cat is like a mottled mix, we call them tortoiseshell, and when they have patches of orange and black and white, they're called calico. Okay, so we are not going to look at tabby cat, and we're not going to look at this whole thing of pattern of distribution. We're just going to look at here at black cats, orange cats, and calico cats. Okay, so we're definitely not looking at the full spectrum of cat genetics for coat color. Okay, so the gene for fur color, whether or not it's the orange or the black or the calico, is on the X chromosome. Okay, so that means it is sex-linked, and the gene is on the X chromosome and not on the Y chromosome. Okay, so if that's not ringing bells, please go back and review the genetics, the basic genetics lecture when I talked about X-linked or sex-linked traits. Okay. So for this fur color gene that's on the X chromosome, there are 2 alleles. So remember, when we're talking about sex-linked or X-linked genes, we use this notation where we have the X and then the superscript for the alleles. Okay. So in this case, the dominant allele is orange fur, which I've represented with a big F. And I chose F because you can easily tell the difference between an uppercase F and a lowercase f. Whereas if I picked O, which might be the obvious example because we usually pick a letter to represent the dominant allele. If I picked O, it's really hard to tell whether you're writing an uppercase or a lowercase. Okay. So X superscript F, big F, is orange fur, and F superscript little f is black fur. Okay. So there's actually 5 phenotypes for fur color when we consider both the color of the fur and the sex of the cat. And this matters because we're talking about a sex-linked trait, right? When we're talking about sex-linked traits, we end up talking about what happens, for males versus females. Okay, so there can be both black males and black females. There can be orange males and orange females, and then there can be only calico or tortoiseshell females. Okay, so these are the 5 phenotypes: black male, orange male, black female, orange female, or females that are calico or tortoiseshell. Okay, so we're going to talk about how this happened. How do we have 2 alleles that give rise to 5 different phenotypes? Okay, so I just wrote this here as a reminder what our alleles are, what the notation for our alleles are. Okay, how this is it's a combination of a sex-linked trait and something called X inactivation. Okay, so let's just think about this systematically. So males have only 1 copy of the fur color gene, right? Because it's sex-linked, and if it's sex-linked, it's not on the Y chromosome, just on the X. Okay. So males will have just 1 copy, and they, one option would be that their copy is X little f, and their Y doesn't have any one of them. Okay, this would yield black male cats. Okay, or they could be X big F, and that would yield orange cats, orange male cats. Okay. Females, on the other hand, have 2 copies of this gene. Okay, so they might have 2 X chromosomes that both carry the little f allele, and that would give you a black cat. Or they could have 2 copies of the big F allele, and that would give you an orange cat. And then here's the interesting one. If they are heterozygotes, and they have 1 copy of the big F and 1 copy of the little f, you then get the calico cats. And this is where the X inactivation is important. So what is X inactivation? So X inactivation is what happens in early embryonic development, and by the way, this happens in humans as well. In every cell, 1 X chromosome is inactivated and it becomes something called a bar body. It's basically super packaged DNA that is no longer expressed. Like, it's so packaged up, it just forms this little blob, and the genes on it are never read. Okay? Once an X is inactivated in a cell, all of the descendants of that cell will have the same X inactivated. Okay, so the point of why this happens is so that every cell has only 1 copy of the X chromosome, because those genes that are on the X chromosome, you're supposed to, most of them work by just having 1 copy. And if you had 2 copies, it would be too much of it. Okay, so we see the effect of this in calico cats or in tortoiseshell cats. Okay, so tortoiseshell cats or calico cats, I'm going to use those terms interchangeably, even though, you know, for a cat fancier there might be a difference. Both of them have patches of black and orange, and their genotype would be X big F, X little f, right? So in the embryo, both of these alleles are present. In early embryonic development, when the embryo is just a cluster of cells, about half of those cells will have the black or the X little f, and the X chromosome with the little f inactivated, and only the orange one would remain active. Okay? Those cells then continue to divide and would give rise to the orange patches on the cat. Okay? In approximately half of the other cells, the opposite happens. The X carrying the orange allele, or the X big F, is inactivated, and only the X little f black allele is active, and that yields these patches of black. Okay. Remember, to get a calico, it's the same kind of thing, but there's another gene that's interacting with this gene that controls the distribution of these pigment cells. So don't worry too much about that. Okay, so actually this X inactivation happens in all female cats. Okay, we just only see the effect of it when we look at the calico cats. Okay, so a female cat that is homozygous recessive for the black fur allele will have in about half of her cells 1 of these X chromosomes inactivated, and half of the other cells the other X chromosome inactivated. But we can't see, even though that happens, we can't see any effect of it because all of the cells will still have the X little f or the black fur allele activated. Okay, the same thing goes for a homozygous dominant female cat who has 2 copies of the big F orange allele. In half of her cells, 1 of these chromosomes will be inactivated, while in the other half, the other X chromosome will be inactivated. But again, we don't see the effect of that. We only see the effect when it's a heterozygous individual, right? Because then in half of the time, you'll get only the X big F, and the other half you'll get only the X little f, and that leads to this patchiness. And that is it for our tour through more complex genetics.